The coelacanth. It was December 22, 1938. A South African museum curator named Marjorie Courtenay-Latimer was sorting through the catch hauled up by a local fishing trawler near the mouth of the Chalumna River. She almost missed it. But something at the bottom of the pile stopped her. A fin. Blue and iridescent, attached to a fish unlike anything she had seen before. She made a sketch and sent it to the nearest ichthyologist she could reach, a man named J.L.B. Smith. His reply, when it came, was barely coherent with excitement. The fish in her sketch had been dead for 65 million years. This is the coelacanth. And the problem with it isn't just that it survived. The problem is that it survived without changing. The fossil record places the coelacanth's body plan in the early Devonian, approximately 400 million years ago. That is before the first trees grew on land, before insects existed, before the dinosaurs were even a distant evolutionary possibility. And the fish Courtenay-Latimer pulled from that trawler's catch in 1938 was, in every meaningful structural detail, the same animal. The same hollow spine. The same lobed, limb-like paired fins that move in a trotting gait, the way land animals walk, alternating opposite limbs in sequence. The same intracranial joint that allows the upper skull to swing upward while feeding, a feature found in no other living vertebrate on Earth. The same rostral organ in the snout for detecting the electrical fields of prey in total darkness. None of it updated. None of it revised. 400 million years, and the coelacanth's response to evolutionary pressure was apparently silence. A second living species, Latimeria menadoensis, was found in a fish market on the Indonesian island of Sulawesi in 1998 by biologist Mark Erdman. It lived 10,000 kilometers from the first known population. Both species in the same body, in the same form, separated by an ocean for millions of years. A 2013 genome study published in Nature, led by Chris Amamiya and colleagues, confirmed what the fossils had suggested. The coelacanth has evolved more slowly at the genetic level than any other vertebrate sequenced. Its protein-coding genes change at roughly 1/3 the rate of tetrapods. The molecular clock is running. The body simply refuses to move with it. Evolutionary biologists have an explanation for this. They call it stabilizing selection. When a body plan is already optimal for its environment, mutations that alter it are penalized. The organism that changes dies before reproducing. The deep-water habitat the coelacanth occupies, between 150 and 700 meters in the Indian Ocean, has remained broadly stable across hundreds of millions of years. There was no pressure to update the design. So the design wasn't updated. It is a reasonable explanation. It is the kind of explanation that satisfies most textbooks. But the Amamiya genome study complicates it in a way that is difficult to dismiss. The coelacanth's genes are mutating. The molecular machinery for change is present and functioning. The mutations are simply not expressing themselves in the body. That is not an organism settling into a comfortable niche. That is something closer to a suppression. Something in the coelacanth's developmental architecture appears to be intercepting change before it can reach the physical form. Some researchers in evolutionary developmental biology have begun asking whether certain body plans can achieve a kind of lock where the embryonic programming becomes so deeply canalized that no viable mutation can find a foothold in the structure. The coelacanth's lobed fins, the very features that link it to the ancestor of all land vertebrates, suggest something stranger still. This animal is not a failed experiment. It is the original blueprint. Every four-limbed creature that has ever walked, climbed, or crawled on this planet carries a version of that fin structure in its evolutionary memory, which raises a question that nobody has quite answered. Is the coelacanth failing to move forward? Or is it the starting point that the rest of life moved away from, and somehow, impossibly, it is still here? There are fewer than 1,000 individuals estimated to survive in the Comoros population alone. The species that outlasted the dinosaurs may yet be outlasted by us. From the deep ocean to the shoreline, what comes next has been sitting on the beach the entire time, in plain sight for longer than the Atlantic Ocean has existed. The horseshoe crab. Every May, something ancient happens on the beaches of Delaware Bay. 500,000 horseshoe crabs drag themselves out of the Atlantic surf and onto the sand to lay their eggs. They do this at high tide, under a full moon, the same way they have done it since before there were forests on land, since before there were insects to fill those forests, since before the Atlantic Ocean itself had finished opening up between the continents. The horseshoe crab does not know it is 450 million years old. It just knows it is time to go to the beach. The horseshoe crab is not actually a crab. It is more closely related to spiders and scorpions, a chelicerate arthropod whose lineage was established in the Ordovician period, roughly 450 million years ago. In 2008, David Rudkin and colleagues at the Royal Ontario Museum described Lunataspis aurora from deposits in Manitoba, Canada, dating to 445 million years ago. It was a horseshoe crab. Near identical in its hinged carapace, its compound eyes, its 10 legs, its long tail spine, its book gills arranged in flat overlapping plates along the underside. The animal that comes ashore in Delaware Bay every spring is, structurally, the same creature that navigated the Ordovician Sea. What makes this remarkable is what that span of time contains. It contains 4 of the 5 great mass extinction events in Earth's history. It contains the Permian-Triassic extinction, 251 million years ago, the worst catastrophe in the history of animal life, when an estimated 81% of all marine genera were erased from the fossil record. The horseshoe crab crossed that event without a detectable structural revision. Its compound eyes, which were old when the Permian began, are still old in the same way now. And then there is the blood. Horseshoe crab blood is blue, colored by copper-based hemocyanin, rather than the iron-based hemoglobin that makes ours red. But the color is not the remarkable part. The remarkable part is what the blood does when it encounters bacterial contamination. It coagulates instantly and specifically around endotoxins produced by gram-negative bacteria. This response, mediated by a compound called limulus amebocyte lysate, is so sensitive and so precise that it became the global standard for pharmaceutical safety testing. Every injectable drug approved by the United States Food and Drug Administration must be tested using horseshoe crab blood. The animal that survived the Permian extinction is at this moment keeping the human species alive. Evolutionary biologists explain the horseshoe crab's stasis as a function of environmental consistency. Shallow estuarine and coastal marine habitats have provided a stable selective backdrop across hundreds of millions of years. The horseshoe crab's generalist diet, tolerating low oxygen water, feeding on worms and mollusks and algae, means it has never been boxed into a narrow ecological corner that would demand structural innovation. There was never enough pressure to force the design to change. So it didn't. But the Permian extinction didn't spare estuarine environments. It collapsed ocean chemistry globally. It crashed oxygen levels. It rewrote marine ecosystems from the bottom up. The horseshoe crab crossed that threshold unchanged. Environmental stability is not a sufficient answer for surviving the worst planetary catastrophe in the fossil record without a single structural modification. 4 living species survive, split across 2 oceans, the Atlantic Limulus polyphemus and 3 Indo-Pacific species, geographically separated for tens of millions of years, near identical in form across that entire span. Some researchers in evolutionary developmental biology have proposed that the horseshoe crab may represent an extreme case of what they call developmental constraint, a body plan so deeply embedded in its embryonic programming that viable mutations simply cannot gain traction against it. The architecture resists change, not because change never comes, but because the system has evolved to reject it before it reaches the body. What the horseshoe crab is telling us, quietly and from 450 million years of evidence, is that some designs are not improved by time. They are only confirmed by it, from the Atlantic coastline to a windswept island at the bottom of the Pacific. What we encounter next is stranger still, an animal that is evolving faster than almost anything alive, while its body refuses to move at all. The tuatara. On Stevens Island, a small wind-stripped rock off the northern coast of New Zealand's South Island, a reptile sits in the dark. It does not move much. It does not need to. Its metabolism runs so slowly that it digests a meal over several days. It is most comfortable at temperatures that would render most reptiles completely lethargic. And it has been here, in this body, in essentially this form, since the Triassic period, approximately 250 million years ago, when the dinosaurs were just beginning to find their footing on a very different Earth. The tuatara, Sphenodon punctatus, looks like a lizard. It is not a lizard. It is the sole surviving member of the order Rhynchocephalia, a lineage that diverged from lizards and snakes before the age of the dinosaurs and once spread across the globe in over 30 recognized genera. Every other rhynchocephalian went extinct. The tuatara, on an isolated island chain at the edge of the world, did not. German biologist Johann Georg Fischer formally established this in 1867, but the deeper strangeness of the tuatara's situation didn't fully emerge until the genomic era. In 2008, a study led by David Lambert and colleagues at Griffith University, published in Trends in Genetics, compared ancient DNA extracted from museum specimens up to 8,000 years old against modern tuatara populations. What they found stopped the team. The tuatara has the fastest mitochondrial DNA evolution rate ever recorded in any animal. It mutates faster than a mouse, faster than a rabbit, faster than a human, by a significant margin. And yet, placed next to its Triassic fossil relatives in the museum drawer, a trained paleontologist would struggle to point to a meaningful structural difference between them. The tuatara's genome is running at full speed. Its body is not moving at all. The structural details are precise. The tuatara has 2 rows of upper teeth that interlock with a single lower row in a shearing motion found in no other living reptile, the same dental arrangement visible in rhynchocephalian fossils from 200 million years ago. It possesses a parietal eye on the top of its skull, a genuine third eye with a lens, a retina, and a nerve connection to the brain, visible through the scales in hatchlings before it is covered over in adults. No confirmed function has been established for it in living animals. It remains literally a window from the outside world directly into the skull, present in the Triassic relatives, present today, with its purpose unknown. The tuatara also remains active at temperatures as low as 7 degrees Celsius, a cold tolerance that is unique among living reptiles and that may have helped it survive New Zealand's climatic swings across the millennia. Evolutionary biologists explain the paradox by separating the molecular clock from the morphological clock. They run independently, the argument goes. A fast mutation rate at the mitochondrial level doesn't necessarily produce structural change if those mutations are selectively neutral, not affecting proteins in ways that alter the body. The genome drifts. The body doesn't have to follow. It is a technically coherent position. But 250 million years is an extraordinarily long runway. If the tuatara's genome is accumulating mutations at a rate faster than almost any animal on Earth, the mathematical probability of at least some of those mutations producing a viable structural novelty across that time span should not be zero. Evolutionary developmental biologists have begun asking whether the tuatara represents an extreme case of what is sometimes called developmental canalization, where the embryonic pathway to a given body plan becomes so deeply grooved that virtually no mutation can redirect it. Others ask the harder question: if an animal can carry the fastest mutation rate in the animal kingdom for 250 million years and still produce the same body, what is the mutation for? Is it possible those changes are doing something else entirely, something we haven't identified yet, something that has nothing to do with the shape of the animal at all? The total wild population stands at approximately 50,000 individuals, confined almost entirely to predator-free offshore islands after near extinction caused by introduced rats and cats, the last survivor of an entire reptilian order mutating furiously, going nowhere, from this cold island to the open ocean, because the next species didn't just outlive its era. It outlived the concept of eras altogether. The shark. The shark existed before trees. Take a moment with that. When the first sharks moved through the ancient ocean, the land was bare rock and shallow pools. There were no forests because trees had not yet evolved. There were no insects. There were no bones in the no insects. There were no bones in the terrestrial world because nothing with bones had yet left the water. And in that pre-forest, pre-insect, pre-bone world, the shark was already there, already hunting, already recognizable, already essentially the thing that is still circling the reef today. The shark lineage is established from dermal denticles, small tooth-like scales, recovered from marine deposits in Nevada and China, dating to approximately 450 million years ago in the Silurian period. By the Devonian, roughly 400 million years ago, sharks had diversified broadly enough that the period is sometimes called the Age of Fishes. The oldest near-complete shark specimen, Doliotis problematicus, from the Campbellton Formation of New Brunswick in Canada, dates to approximately 409 million years ago and already displays the cartilaginous skeleton and the fundamental body plan that defines modern sharks. Randall Miller and colleagues identified it as the earliest recognized shark with both teeth and paired fins preserved together, confirming the full structural package was already present before the first land vertebrates existed. Since then, the shark has survived every mass extinction event on record, 5 of them. The one that ended the Permian, the one that ended the Triassic, the one that ended the Cretaceous. The asteroid that killed every non-avian dinosaur 66 million years ago, an event that eliminated approximately 75% of all species on Earth, produced no meaningful revision to the shark body plan. The torpedo profile. The cartilaginous skeleton, lighter and more flexible than bone, a genuine engineering advantage for an open water predator. The multiple rows of continuously replacing teeth. The ampullae of Lorenzini, electroreceptive pores around the snout capable of detecting electrical fields as faint as 5 nanovolts per centimeter, present in fossils hundreds of millions of years old, structurally unchanged. The lateral line, running the length of the body, detecting pressure differentials in surrounding water, documented in 300 million-year-old Carboniferous specimens in the same architectural form it takes today. Evolutionary biologists call this a case of extreme adaptive success. The design reached near optimum efficiency early and has been maintained because any deviation from it produces a less competitive animal. Cartilage over bone is genuinely superior for an active pelagic predator. Continuous tooth replacement means the shark is never disadvantaged by dental wear. The body plan won the competition early and was rewarded for not changing. That is how stabilizing selection is supposed to work. But a 2014 study in BMC Evolutionary Biology examining shark genomics found something that complicates the clean narrative. Genes associated with DNA repair, immune response, and tumor suppression showed signatures of strong positive selection in sharks, meaning the genome is not static. It is actively maintained and updated. The molecular machinery is clearly working. The structural output simply hasn't changed. The shark's genome is being refined, generation by generation, in ways we are only beginning to understand. What is being refined is not the shape. It is something underneath the shape. The great white can detect blood at concentrations of 1 part per million across distances of over 400 meters. It hunts with sensory architecture that was old before the first flower bloomed. It has crossed every extinction event in the fossil record and emerged each time as the same animal. Most evolutionary narratives describe progress, refinement, specialization, the accumulation of novelty. The shark is the counter-argument. It says that in 450 million years of relentless selective pressure, nothing has improved on this design, not once. Which raises the question some researchers have started asking out loud. Are there physical and mathematical limits to what a body plan in open water can be? And has the shark simply reached them? From the open ocean to the deep sea floor. What comes next chose a different strategy entirely, not speed, not power, but a design of mathematical elegance so precise that Renaissance artists would spend centuries arguing over whether it was divine. The Nautilus 500 million years ago, while your ancestors were soft-bodied worms navigating the shallow Cambrian Sea, a creature with a coiled shell and grasping tentacles was already doing what it does today. It was descending at night, rising before dawn, navigating by pressure and chemistry through water so deep and cold that most animals cannot survive there. It has been performing this cycle, this same daily vertical migration, in this same body for longer than any vertebrate lineage has existed on Earth. The nautiloid lineage traces to the late Cambrian, approximately 500 million years ago. Early forms like Plectronoceras already carried the fundamental architecture that defines living nautiluses today. An externally chambered shell, a siphuncle tube threading through every internal chamber to regulate buoyancy by adjusting the ratio of gas to liquid, and a soft body that withdraws entirely into the shell when threatened. Over the following hundreds of millions of years, nautiloids diversified enormously, producing thousands of fossil species in coiled, straight, and curved forms that dominated the world's oceans for much of the Paleozoic era. Then came their cephalopod relatives, the ammonites, who diversified brilliantly across the Mesozoic in hundreds of genera with elaborate shell ornamentation and complex suture patterns. The ammonites evolved, they competed, they filled niches, they were everywhere. The Cretaceous-Paleogene extinction 66 million years ago eliminated every single ammonite species. It was total. The Nautilus, morphologically conservative, structurally unchanged, descended into deeper water and continued its nightly migration as if nothing had happened. Peter Ward's fieldwork in the Pacific, documented across decades of research, established that living Nautilus pompilius descends to depths of at least 700 to 800 meters at night, exposing the shell to pressures exceeding 23 atmospheres, then rises to between 100 and 300 meters by day, a daily vertical commute of 600 meters or more through water that ranges from near freezing to tropical. The shell handles this pressure cycle without fracturing because of its internal chambered architecture, a system already documented in near-identical form in Ordovician fossils 450 million years old. The Nautilus eye is a pinhole camera. There is no lens. There has never been a lens. This is, by conventional evolutionary logic, a puzzle. Lens-based eyes have evolved independently dozens of times in the history of animal life. They provide dramatically sharper vision than a pinhole aperture. Insects evolved compound lenses, vertebrates evolved camera-type lenses, cephalopods like octopuses and squids evolved eyes that are, structurally, almost identical to human eyes. Yet the Nautilus, which shares the same ancestral cephalopod lineage as the octopus, has maintained a pinhole eye for 500 million years without ever producing a lens. Evolutionary theory predicts that useful innovations, sharper vision in particular, are strongly selected for. The Nautilus has had 500 million years to develop one. It has not. Scientists explain both the lens absence and the morphological stasis through a combination of deep water refuge and metabolic efficiency. The Nautilus can survive months without food by dramatically reducing its metabolic rate. Its simple eye is adequate for finding food and avoiding predators at the depths it frequents. The lack of sophisticated sensory demands in its niche means there is no pressure towards sensory refinement. Environmental constancy at depth provides the stable selective backdrop that stabilizing selection requires. Each explanation, taken alone, is coherent, but taken together they describe something harder to dismiss. The Nautilus was present when the ammonites rose and conquered the Mesozoic ocean. It watched them diversify into hundreds of forms. It was still there when the asteroid arrived and wiped every one of them from the fossil record. The animal that refused to innovate outlasted the animals that built an empire of innovation. Some evolutionary theorists have started using the word they were previously reluctant to use. They call it optimization, a design so complete that the standard mechanisms of evolutionary change have no profitable direction to push it. Others ask the harder version. If 500 million years of selective pressure cannot improve a pinhole eye, what exactly is the Nautilus's eye actually for? From the depths of the Pacific to the surface of a desert puddle. What comes next doesn't survive by being ancient. It survives by refusing to exist at all. The horseshoe shrimp. In the Sahara Desert, when it rains, which is almost never, certain dry basins flood briefly before the water evaporates back into the sky. In the hours after the flood, something hatches from the dust. A small, three-lobed crustacean, armored across its back with a fan of legs and two bulging compound eyes, pulls free of its casing and begins to swim. It feeds. It grows. Within 2 weeks, it is an adult, laying eggs of its own. Then the basin goes dry again. The adult dies. The eggs fall into the sediment and wait. They may wait a year. They may wait 10 years. In one documented laboratory study, eggs remained viable for 27 years in dry storage and then hatched when water was added. The animal inside those eggs has been doing this since the Triassic period, approximately 220 million years ago, when the first dinosaurs were just appearing on land. This is Triops cancriformis, the European tadpole shrimp, sometimes called the horseshoe shrimp, and it holds a distinction that no other animal can match. Fossils from the Kuiper Formation of Velberg, Germany, dated to approximately 220 million years ago, are morphologically indistinguishable from living specimens. German paleontologist Carl von Siddel recognized this in the 19th century when he compared museum fossils to living animals and found the match so complete he could barely justify separate descriptions. Triops holds the record, contested in some corners of paleontology, but supported by the morphological evidence as the oldest confirmed living animal species on Earth. The key to its survival is a biological mechanism that should not be possible at the scale it operates. When a Triops pool dries up, the embryos enter cryptobiosis, a near-total suspension of metabolic activity. During desiccation, the embryos produce a sugar called trehalose, which replaces the water in their cells, preventing the ice crystal damage that would otherwise destroy them. Metabolism drops to levels that cannot be meaningfully measured. Time, in any biological sense, stops. The eggs are not waiting for rain. They are not, in any functional sense, experiencing the wait at all. When water returns, a chemical signal triggers resumption of development, and the animal picks up from where it paused, potentially decades later. This mechanism is shared, independently evolved, with tardigrades and certain desert plants. In Triops, it may carry a deeper evolutionary consequence than previously recognized. Each generation that hatches is, in a sense, hatching from the same ancestral moment. The normal accumulation of generational change, the steady drift of the genome through time, is disrupted by a life history where entire decades between reproductions can be spent in a state outside time. Some researchers have theorized that in extreme cryptobiotes, the standard relationship between time elapsed and genetic change simply breaks down. The organism is not passing through evolutionary time in the way that continuously reproducing species do. Evolutionary biologists explain Triops stasis through the boom and bust demands of ephemeral pool ecology. The core challenge has not changed in 220 million years: survive desiccation, hatch fast when water appears, reproduce before it evaporates. Any body plan that solves that problem reliably is strongly favored, and any deviation from it is penalized. Stabilizing selection in a volatile but structurally unchanging niche. It is the same explanation applied to other entries on this list. But 220 million years is a span that contains the Triassic-Jurassic extinction, the breakup of Pangaea, the entire rise and fall of the dinosaurs, and multiple glacial cycles that redistributed fresh water across every continent. The pools changed location. The continents moved. The climate swung between extremes that reshaped entire ecosystems. Triops kept using the same body to solve the same problem in all of them. Populations exist on every continent except Antarctica. They live in Saharan rain basins, rice paddies in Southeast Asia, mountain pools in Japan, desert vernal pools in the American Southwest. A 2005 molecular study found surprisingly low genetic diversity across populations despite their enormous geographic spread, consistent with extreme morphological conservatism across millions of years of reproductive isolation. The animal is everywhere, and everywhere the same. What Triops may be demonstrating is something evolutionary theory hasn't fully accounted for. That when an organism can stop time, it may also stop the biological clock that drives everything else forward. From the desert floor to the rainforest undergrowth. What comes next has been crawling through wet leaf litter since the Cambrian. 500 million years old. Soft-bodied, ancient, and carrying in its strange unjointed legs a structural secret that connects it to the greatest evolutionary explosion in the history of animal life. The velvet worm. In a rainforest in New Zealand, in the deep wet dark under a rotting log, something is hunting. It moves on stubby, unjointed legs that fill with fluid pressure to extend and retract between 14 and 43 pairs of them, depending on the species. Its body is soft and damp, covered in rows of sensory tubercles. From two openings flanking its mouth, it fires jets of adhesive slime that harden on contact, immobilizing prey that can be several times its own size. Then it moves in and begins feeding. Paleontologists have found this animal in the Burgess Shale of British Columbia, frozen in 500-million-year-old rock from the Cambrian explosion. It looks the same. The velvet worm belongs to the phylum Onychophora, and its evolutionary position is one of the most significant in the animal kingdom. It occupies the living bridge between annelid worms and arthropods, carrying features of both lineages simultaneously in a body that has not been fundamentally reorganized since the Cambrian. Its fluid pressure legs, called lobopods, are documented in Cambrian fossils, including a Shia pedunculata, described by Simon Conway Morris in his landmark analyses of Burgess Shale fauna. A 2014 paper by Javier Ortega Hernandez and colleagues in Nature re-examined the Cambrian lobopodian hallucigenia and confirmed its position in the stem lineage leading to living velvet worms, establishing a nearly unbroken morphological thread from 500 million years ago to the present. The adhesive slime mechanism is unlike anything else in the animal kingdom. Fired from oral papillae at speeds exceeding 4.5 meters per second, accurate to within centimeters, it is a predatory technology present in fossil relatives and unchanged in living species. Approximately 200 living species are recognized, distributed across tropical and subtropical forests in South America, Sub-Saharan Africa, Southeast Asia, and Australasia. A 2014 molecular clock study by Oliveira and colleagues confirmed that velvet worm divergence events track the breakup of the supercontinent Gondwana, which began approximately 180 million years ago. The lineage had already achieved its modern body plan before the continents separated, and then sat on each departing landmass and went along for the ride, unchanged, as the oceans opened between them. What makes this not just remarkable but genuinely difficult to explain is what the velvet worm's relatives did next. The arthropods, sharing the same Cambrian ancestor, sharing the same starting point, went on to produce beetles, butterflies, crabs, spiders, centipedes, shrimp, barnacles, and somewhere between 5 and 10 million other species in what is arguably the greatest evolutionary diversification in the history of animal life. They are the most species-rich animal group on Earth. They are in every environment on the planet. They did all of this by innovating relentlessly from the same lobopod body plan that the velvet worm inherited alongside them and never modified. Evolutionary biologists explain this divergence through what they call developmental constraint. The lobopod body plan, fluid pressure legs attached to a soft, flexible trunk, is functional but has no obvious structural pathway toward the jointed limbs and hardened exoskeleton that enabled arthropod diversification. The transition requires a specific suite of genetic and developmental changes, a toolkit that the arthropod ancestor acquired and the velvet worm ancestor apparently did not. Once that toolkit was absent, innovation in the velvet worm's direction was closed off. It stayed in the corridor it was already in. But staying in a corridor for 500 million years, across the breakup of 2 supercontinents, through multiple mass extinction events, while everything around you either innovates wildly or goes extinct, requires its own kind of explanation. Some Evo-Devo researchers have proposed that the velvet worm's developmental architecture represents not just constraint, but something more like a biological equilibrium point, a configuration so energetically stable that no mutation arriving at the embryonic level can gain traction without destroying viability. The system resists perturbation not because change would be disadvantageous, but because the developmental logic of this body plan has no tolerant pathways for change to travel. 200 species across 6 separated landmasses descended from a common ancestor 500 million years ago, all in the same body, all hunting with the same slime. The arthropods took the same beginning and built 10 million species out of it. The velvet worm looked at all of that possibility and stayed exactly where it was. From the forest floor of Gondwana to the wetlands of eastern Australia. Because what comes next didn't just refuse to choose one direction, it chose all of them simultaneously for over 100 million years without arriving anywhere coherent. The platypus. When the first dried specimen of a platypus arrived in Britain from Australia in 1799, the leading naturalist who examined it inspected the bill carefully, then looked for stitches. He was certain someone had sewn a duck's bill onto a beaver's body as an elaborate prank. He was wrong. But his instinct was, in a way, scientifically understandable. Because a platypus is a mammal that lays eggs. It has the bill of a duck, the tail of a beaver, the venomous ankle spurs of a reptile, and the electroreception of a shark. It also has no stomach. Its esophagus connects directly to its intestine. When the platypus genome was sequenced in 2008 and published in Nature by Wesley Warren and colleagues, the team described it as resembling, in their own published language, a mosaic of reptile and mammal features that fundamentally challenges our understanding of vertebrate genome evolution. They had expected a relatively simple picture. They got something that required a new vocabulary. George Shaw formally described the platypus as Ornithorhynchus anatinus in 1799, and Johann Friedrich Blumenbach named the genus the following year. The broader monotrem lineage, the group containing platypuses and echidnas, diverged from all other mammals approximately 166 million years ago in the mid-Cretaceous, making it older than the asteroid that ended the dinosaurs. Fossil relatives have been found in Patagonia, Argentina, in deposits approximately 61 million years old, confirming that the platypus's immediate ancestors were once nearly global before Australia's isolation left them stranded on a continent increasingly free of placental competition. The genome is the central mystery. The 2008 Warren study found that the platypus carries genes for vitellogenin, the protein used to build egg yolk, which it shares with reptiles and birds, alongside the casein genes that produce mammalian milk. Both systems simultaneously in one animal. Its sex determination system involves 10 chromosomes, 5X and 5Y in males, an arrangement found in no other known vertebrate. Those chromosomes share ancestry with both the mammalian XY system and the bird ZW system, suggesting they are the layered remnant of multiple ancient lineages whose genomic logic has never been fully resolved in one direction or another. The platypus also has no genes for pepsinogen, the precursor to the primary enzyme in stomach acid. The stomach, which most vertebrates have maintained since the Devonian, is simply absent. Food passes from esophagus directly to intestine, a condition shared only with certain ancient fish lineages. The electroreception in the bill is achieved through approximately 40,000 electroreceptors and 60,000 mechanoreceptors, allowing the platypus to hunt with its eyes, ears, and nostrils sealed shut, detecting the electrical signals produced by the muscle contractions of freshwater invertebrates buried in riverbed sediment. This system evolved independently from the shark's ampullae of Lorenzini. Two completely unrelated lineages, separated by hundreds of millions of years of evolution, converged on electroreception independently. The platypus reinvented a solution the shark had already been using for 450 million years. Evolutionary biologists describe the platypus as a mosaic organism, an animal in which different body systems have evolved at different rates under different selective pressures, retaining ancestral characters in some systems while innovating in others. Egg-laying may have been retained because live birth offers no strong advantage for an animal that incubates eggs in a protected burrow. each retained primitive feature has a functional explanation. The framework is reasonable. But the platypus isn't simply retaining old features, it is simultaneously running reptilian egg-laying and mammalian lactation. It evolved entirely new electroreception convergently with a shark. It carries a chromosomal architecture that resembles 3 different vertebrate lineages stacked on top of each other. It has been doing all of this at once in the same body for over 160 million years, without resolving any of it into a cleaner, more unified design. Some evolutionary geneticists have proposed that the platypus genome represents deep time mosaicism, where different genomic regions are essentially running separate evolutionary programs, never integrated into a single coherent direction. The platypus may be less a unified evolutionary story than a collision of several, none of which has won. The question some researchers are now asking is not why the platypus looks so strange. The question is whether the platypus's genome has simply never received the organizing pressure required to make it resolve, and whether 160 million years without that pressure is itself the explanation. From the wetlands of Australia to the riverbanks of the ancient world. Because what comes next didn't refuse to choose between directions. It chose no direction at all, and it watched every other choice get destroyed. The crocodilian. 95 million years ago, a crocodilian lay on the bank of a river in what is now North Africa. Above it in the river, a Spinosaurus 4 times its length was hunting. Around it, other dinosaurs moved through the heat. The Spinosaurus is gone. Every non-avian dinosaur is gone. The pterosaurs are gone. 75% of all species on Earth that were alive 66 million years ago are gone. The crocodilian is still on the riverbank. It has not updated its design since before the asteroid fell. It is waiting with a patience that is difficult to comprehend from the outside. The modern crocodilian lineage, the order Crocodilia, is established from approximately 95 to 84 million years ago in the late Cretaceous. Steve Salisbury and colleagues described Isisfordia duncani from Queensland, Australia in 2006 as one of the earliest confirmed true crocodilians, already displaying the armored scutes, the laterally flattened tail, the elevated eyes and nostrils, the palatal shelf that allows breathing with the mouth submerged, and the overall ambush predator architecture that defines every living species today. The broader Archosaurian lineage from which crocodilians descended, the one they share with dinosaurs and pterosaurs, extends to approximately 240 million years ago in the Triassic. When the asteroid hit, every other member of that lineage died. The crocodilian did not change, it survived. The 4-chambered heart is the detail that most stops biologists. Every other living reptile has a 3-chambered heart. Crocodilians have 4 chambers, separating pulmonary and systemic circulation with the same efficiency as mammals and birds. This cardiovascular architecture gives them a physiological performance ceiling that other reptiles cannot reach and enables the explosive bursts of power they deploy in prey capture. The 4-chambered heart is documented in their Archosaurian relatives and represents a feature that was, in crocodilian terms, already ancient when the Cretaceous began. And then there is the immune system. A 2008 study from Louisiana State University, led by Mark Merchant and colleagues, found that crocodilian blood serum demonstrated broad-spectrum antibacterial activity, including documented neutralization against strains of MRSA and, in initial tests, HIV, an immune response significantly more potent than anything produced by comparable mammalian immune systems. The practical implication that a 95-million-year-old body plan may be producing pharmaceutical compounds we cannot yet synthesize is one that ongoing research has not resolved. Crocodilians can survive without food for up to 2 years by metabolizing reserves stored in the tail and reducing their metabolic rate to levels that approach dormancy. Their salt-secreting integumentary sense organs on the tongue, which double as sensory detectors, are present in near identical form in late Cretaceous fossils. Their parental behavior, nest construction, nest guarding, and transport of hatchlings in the parent's jaws is documented in fossil relatives of comparable age, suggesting that even the behavioral complexity of these animals has been frozen for tens of millions of years alongside the body plan. Evolutionary biologists explain crocodilian persistence through ecological generalism and physiological buffering. An ambush predator in fresh and estuarine water, tolerating wide salinity and temperature ranges, capable of extreme fasting, produces an animal with few specialized vulnerabilities. When food webs collapse and niches disappear simultaneously, as they do during mass extinctions, generalist designs with low energetic maintenance costs tend to outlast specialized and innovative ones. The crocodilian's morphological conservatism is, in this view, itself the survival strategy. But here is what makes that argument strange. The crocodilian is more closely related to birds than to lizards. Its closest living relatives diversified, after the same KPG extinction event, into approximately 10,000 species of radically different form, from ostriches to hummingbirds to penguins. The crocodilian, starting from the same post-extinction moment, produced 27 living species in 95 million years, all of them recognizably the same animal. The birds took the identical starting point and built an empire. The crocodilian took it and went back to the riverbank. Some paleontologists have begun calling this a strategy rather than a failure. If morphological conservatism is positively selected for at the geological timescale, if the body plan that refuses to specialize is the one that survives every reset the planet delivers, then the crocodile isn't the animal that couldn't evolve. It's the animal that learned something about time that we are still working out, from the tropical riverbank to the edge of the tidal flat. What waits there is smaller, quieter, and in some ways more impossible than anything before it. A shellfish the size of a fingernail that has survived every extinction in the fossil record since before complex life fully organized itself, and which you can order in a Japanese seafood market today. The Lingula brachiopod. It is the size of a fingernail. It lives buried in mud at the edge of the sea, anchored by a fleshy stalk that pulls it deeper into the sediment when disturbed, extending a fan of tentacles through a narrow gap to filter particles from the water passing overhead. It requires almost nothing. It produces almost nothing visible. It has been doing exactly this since before vertebrates existed, since before jaws existed, since before the Himalayas had begun to rise or the Atlantic had begun to open. If you placed a living specimen next to a Cambrian fossil from 500 million years ago, a trained scientist would struggle to identify a meaningful structural difference between them. This is Lingula, a lingulid brachiopod, and it carries a distinction that no other animal in this video can claim. 500 million years of documented morphological continuity confirmed across fossil deposits on every major continent, anchored by comparative studies stretching back to the 19th century when paleontologist Charles Doolittle Walcott, who would later discover the Burgess Shale, described Lingula fossils from Cambrian deposits and noted with visible unease that they were essentially identical to the animal currently alive in the tidal flats outside. The brachiopods as a phylum were once the dominant animals on the Paleozoic seafloor. They produced roughly 30,000 fossil species across 200 million years of ocean dominance, a diversity comparable to the mollusks that would eventually replace them. Lingula was present at the beginning of that empire. it was present at its collapse. In the Permian-Triassic extinction 251 million years ago, the worst catastrophe in the history of animal life, brachiopod diversity fell from approximately 400 genera to fewer than 20 in a geological instant. The phylum lost roughly 97% of its genera. Lingula crossed that boundary without a detectable structural modification and emerged on the other side into a Triassic ocean almost entirely rebuilt around different animals. A 2016 genomic study of Lingula anatina, published in Nature, found that the gene regulatory networks controlling shell production in Lingula had not substantially reorganized since the Cambrian divergence. Not the genes themselves, but the architecture that controls how those genes are deployed, the developmental wiring beneath the body plan, preserved unchanged across 500 million years of genomic replication. This is a different and deeper form of stasis than simply retaining a useful shape. If the regulatory architecture hasn't been reorganized, the organism structurally cannot generate the kind of developmental variation that morphological change requires. It is not that Lingula refuses to evolve. Its genome may have arrived at a configuration in which meaningful morphological evolution is no longer available as an output. The burrowing mechanism itself is documented in Ordovician trace fossils called lingulicnus that are structurally identical to burrows made by living Lingula today. 450 million years of the same muscle contractions, the same withdrawal reflex, the same anchor and extend behavior preserved in the sedimentary record as a continuous thread. Lingula tolerates extremely low oxygen environments, hypoxic conditions that correspond directly to the ocean anoxia associated with multiple mass extinction events, suggesting its stress tolerance profile is precisely calibrated to survive the conditions that have repeatedly destroyed the biosphere around it. And then there is the detail that gives this entry its sharpest edge. Lingula anatina is sold in fish markets in Japan, China, the Philippines, and New Caledonia. It is consumed as food, sometimes raw, sometimes grilled. The Cambrian ancestor of the animal on your plate survived every extinction event in the fossil record. It watched the brachiopod empire rise and fall around it. It watched the dinosaurs appear and disappear. It filtered mud through 500 million years of planetary catastrophe, and it is still filtering mud in the tidal flats outside coastal cities today, unhurried, unchanged, and apparently unbothered by the fact that the entire history of complex animal life has happened around it without managing to find a single useful improvement to suggest. Evolutionary biologists present the standard answer: estuarine generalism, low metabolic demands, burrowing protection, hypoxia tolerance. Each component is well supported, and none of it is wrong. But taken together, what the evidence describes is an organism that has crossed 5 extinction events by being so fundamentally, completely, architecturally itself that nothing the planet has attempted in 500 million years has found a useful edge against it. The oldest confirmed living animal species, the size of a fingernail, sitting in the mud at the edge of the sea as it has always done, since before the world we know existed. 10 species, 500 million years of evidence, 5 mass extinctions, and in every single case the same outcome. The animals that refuse to change are the ones still here. What connects the coelacanth to the Lingula brachiopod isn't just age. It is a specific quality of resistance, a biological stubbornness that conventional evolutionary theory can partially account for but cannot fully explain when taken as a pattern. We have a name for it. We call it stabilizing selection. When a body plan is already optimal for its environment, the theory goes, change is penalized. The organism that mutates in a useful direction is the exception. Most mutations are neutral or harmful. The design that already works is maintained. That explanation has real scientific merit, and none of the items on this list should make us dismiss it. But stabilizing selection explains individual cases. It does not explain the pattern of 10 animals across 6 continents in radically different environments, across timescales that dwarf the existence of our entire genus, all exhibiting the same refusal. The shark lives in open ocean. The horseshoe shrimp lives in desert puddles. The velvet worm lives in tropical leaf litter. The nautilus lives in the deep Pacific. The horseshoe crab goes to the beach in Delaware. The selective environments could not be more different. Yet all of them converged on the same outcome. A body plan established early, maintained across multiple planetary catastrophes, and apparently immune to the relentless pressure of evolutionary time. What this list is quietly suggesting is that evolution may have a second mode. Not just the accumulation of novelty, not just the branching tree of diversification, but something more like a lock. A configuration in which the genome continues to function, continues to replicate, continues to accumulate mutations, but the body will not follow. The tuatara's genome is evolving faster than almost any animal on Earth. The coelacanth's genes are mutating at a measurable rate. The mutations are not reaching the structure. Something in the developmental architecture of these organisms has become effectively decoupled from time. And we do not yet have a fully satisfying explanation for how that decoupling works, or why it happens in some lineages and not others. What we do know is that the animals which survived everything, the ones that looked at 500 million years of planetary disruption and didn't blink, were not the most innovative. They were not the most specialized. They were not the most aggressive. They were the ones that found a configuration deep enough to resist the pressure that destroyed everything else. Most people encounter these animals in a museum exhibit or a nature documentary and move on. They accept the label. Living fossil? Curiosity? Interesting. The fact that you're still here means you heard something else in the data. You heard the question the label is trying to avoid. Not just how did these animals survive, but what does their survival mean for everything we think we know about the direction of life on Earth? This channel is for the people who can't let that question go. Hit subscribe and make it official. Then go to the comments and tell me, of these 10, which one do you think is the most impossible to explain with the standard model? Pick one and make your case. I want to see the argument. These animals didn't survive by winning. They survived by refusing to play the game that everything else was playing. The shark watched the ocean reinvent itself a dozen times and said nothing. The lingula watched an entire phylum collapse around it and kept filtering mud. The tuatara carried the fastest mutation rate in the animal kingdom for 250 million years and never spent a single one of those mutations on a new body. What they are telling us, if we are willing to listen, is that our model of progress, the idea that forward is always better, that change is always gain, that evolution is always an improvement on what came before, may be a story we invented about ourselves. The oldest things on Earth didn't get old by moving. They got old by knowing exactly when to stay still. The species that refused to evolve outlasted everything that tried.